Thank you so much, Cindy, for the introduction. And I'd like to thank everybody who's present in person and on Zoom for coming to my talk today. Okay. So today I'm going to be talking about kind of an emerging story within my research group that we've been working on for about the past year and a half. And I don't have any disclosures to talk about today. So an outline for my talk is that we're first going to start talking about the lung microbiome, what's the composition of the lung microbiome. Then I'm going to go into some of our research talking about microbial dominance within the pigtail macaque lung. Then I'm going to kind of segue talking a little bit about a specific bacterium called Whippleii and talk about the characteristics of Whipple's disease. Then I'm going to go back to our data talking about different environmental factors that are influencing the lung microbiome, concluding with some of our future directions for the project. But the main takeaway that I want you to take from this talk today is that the pigtail macaque is a really highly relevant and insightful model that we can be using to study the human lung microbiome. So what is thought of as having a healthy human lung microbiome is that we want to have overall high microbial diversity, just like we find within the gut. But overall, the amount of diversity is going to be a lot less than what is seen within the gut. We also want to make sure that there's low microbial density, so overall having low bacterial loads, and wanting to have kind of a uniform spatial distribution of these microbes where they're evenly distributed throughout the lungs. And then in states when we get dysbiosis, we're going to have decreases in microbial diversity, increases in these microbial densities, and we can start to get spatial variations of these different microbes. A good example of this is during TB infection, where you're going to have certain sites where the bacteria is really going to be concentrated. And there can be several different factors that can influence lung dysbiosis. This can include inflammation and different respiratory diseases, smoking, pollution, as well as medications. And there's this idea about how the lung is being seeded, and that the microbes are most likely coming from the oral cavity as well as the oral pharynx. And so we have immigration of these microbes through different mechanisms such as inhalation as well as microaspiration. And then we're also going to have different extinction methods, including cough. You could have immune clearance and mucociliary clearance as well. And so it's really this dynamic that is leading to the constant input of different microbes and then these being expelled. And so in a healthy airway, we're overall going to have more microbes being taken out of the lung than are coming in. And so there are going to be a lot of microbes that are going to be transient, but there can really be some that are constant. And here I'm just highlighting on the left, there are 6 major genera within the human lung that are thought to be the core microbes. There can also be a variety of different factors within the immune environment, of the environment within the lung, including pH and temperature, oxygen, as well as the resiliency of the different tissue within the lung that can also lead to differences in the microbes. But when we kind of go into this disease states, we start to have often issues with the extinction of these different microbes out of the lung. This could be due to dysfunctional cilia or changes in the immune responses, and this is going to lead to certain microbes taking over in the lung. And so on the right, I'm highlighting different types of lung diseases and the types of microbes that are associated with becoming enhanced during those diseased states. And so my lab uses the pigtail macaque for studying infectious diseases. And they get this name from having a curly tail that looks like a pig. And what I'm overall highlighting here in the middle is that these are the different types of human pathogens that we use to model in these animals, as well as this animal model is really highly used for the preclinical testing of vaccines and therapeutics. And in recent years, my lab had an interest of looking at respiratory infections, specifically with SARS-CoV-2. And we started to think about what could be the potential interactions between respiratory infection and the microbiome. So kind of then to first start out, what we wanted to just ask, well, what was actually known about the pigtail macaque lung microbiome? And to our surprise, there was actually no information regarding the composition of the lung microbiome. So what we initially started to do, to take it upon ourselves, was to establish this map of the lung microbiome. And this was really so that we could enhance this model of human infectious diseases. And so what we're able to do is we're able to take different paired specimens from the animals. So we can get bronchial alveolar lavage, or BAL, so that we can look within the lung. We get oral swabs, we get blood, as well as stool. We're also able to collect a variety of different metrics from these animals, including their clinical histories that may include antibiotic usage, their different housing data, where have these animals come from over the years, their different demographics, as well as other different features of these animals. Next, we're able to characterize the microbiome in the lung, as well as in other different sites. And then we have ongoing, what we're looking at is how the microbial composition within the lung is shaping different immune responses locally as well as systemically, with our overall goal of integrating this all together to better understand how different microbial compositions contribute to respiratory health. So far we've collected bronchial alveolar lavage samples from 166 macaques spanning across 3 different species. But what I'm really going to focus on today is on the pigtail macaque, although many of the findings that we've found today we've also seen in the other species. And so previously we had historically unpaired specimens, so these were just bowel samples, and then more recently we started to be collecting contemporary paired specimens of the oral swab, the blood, as well as the stool. And so today I'm going to be highlighting the 81 different samples that we have sequenced so far. And so this is including 10 paired samples as well as 71 unpaired samples. To assess the microbiome, what we do is we take our different samples. We can then extract the DNA. And then in collaboration with Steve Salaponte and the MIMC, we do 16S RNA amplification of the V3, V4 region of the 16S gene. This library then goes to sequencing, and then through a variety of different processes, we can clean up the data, and we can do a QC to do for our data analysis. And when we're talking about studying the microbiome, for those of you who may be less familiar with some of the different metrics that we use, so there's a variety of different diversity metrics. The first is alpha diversity, and so this is going to be diversity within a sample. And so we can measure different things like richness, which is the total number of species within our community. We can measure the evenness, the degree to which different species are equally abundant. And then we have other metrics like Shannon diversity, which factors in both richness and evenness. And then we can look at phylogenetic diversity, that's looking at evolutionary distance. And so kind of a good example on the left with these trees is the top forest would overall have low diversity, whereas the bottom forest would have a lot more, would have higher alpha diversity. We can also be looking at what is called beta diversity, and this is diversity across samples. And these can factor in things like relative abundance and also the phylogeny of the different microbes, but this is so that we can measure compositional changes between our different communities. So now an example on our right with our different forests. So overall, these 2 forests would have the same alpha diversity, but the forest on the right and the forest on the bottom, even though they have the same alpha diversity, their composition is overall different, and so they would be different. And so that's just kind of a way to be understanding the different type of metrics that I'll be presenting today. So when we first wanted to look at the microbial composition of the lung microbiome across our 81 macaques, first starting at the phyla level. So this is showing on the x-axis is our 81 animals, and on the y-axis is the relative abundance of at the phyla level within the lung microbiome. And what I'm highlighting here on the right are some of the top 4 core phyla within the human lung microbiome. And so again, a lot of these microbes that are found within the human lung are also found within the macaque lung. And then if we go to the genus level, again, on the left, I'm highlighting the 6 core genera that are found in the human lung, and these are also found within the pigtail macaque lung. But now when we look at the genus, what we can really quickly notice is that there is a singular genus that is really quite prevalent and dominant within our animals. So overall, this is the genus Tropherma, and overall our animals have over 80% carriage of this bacteria within their lung, and about 2/3 of animals are dominant. So that means that over 50% of the relative abundance of the microbes within the lung are this single genus. And if you look at the upper left-hand corner of this graph, you can see that we even have a handful of animals where over 90 and even over 99% of their lung microbiomes are this single genus. And it's no surprise that if we have what we're calling Tropherma dominance, that overall these animals are going to have low microbial diversity. And so this is kind of going against what we think of as being a healthy lung. And I should note that all of these animals are just random ones from our colony, and so these are all considered healthy macaques. When we also look at the microbial, the community structures in these different animals, what I'm showing now here is that we have tropherma-dominant animals are in the black, and that they are, they're segregating separately from animals that are non-dominant or those that do not have tropherma. So this is really a unique community structure of the microbial composition of these tropherma-dominant animals. So now we're just going to take a little bit of a detour away from the data, because now you're probably wondering what exactly is tropherma. So currently there's only one characterized species for tropherma, and this is Tropherma whippli. And because we're doing this at 16S, we don't have enough resolution to go down to the species level. So at this point, we're assuming that what we are identifying in our animals is Tropherma whippli. So Tropherma whippli is a gram-positive bacteria. It's part of the class Actinomycetes, which is thought of more of being a ubiquitous environmental bacteria. It's a rod-shaped bacteria, and what it's more classically known is for causing a disease called Whipple's disease that more classically presents as a gastrointestinal infection. And what happens is it infects macrophages. It then gets this phenotype of having what are called foamy macrophages. And if this goes untreated, it causes a wasting syndrome that eventually leads into death. But what is more not well widely known is that this bacteria can actually have wide tropism. It can go into the CNS, it can go into the heart and cause endocarditis. It can also go into the lung and cause different lung complications, and can also cause a pneumonia, and so it's called Whipple's pneumonia. So overall carriage within the stool during the gastrointestinal infection can be in about 1 to 10% of individuals, but it happens to be more common in individuals that work in sewer plants or on farms. And the gut infection is actually more commonly found in older white males. So T. whippli gets its name. It was first discovered by George Whipple back in the early 1900s. And then it wasn't until the early 1990s when it first got its first name, when the partial sequence was identified. But then it wasn't until later in the 2000s when they hold the full genome, and then they decided to rename it to be more closely related to George Whipple's name. But because it was very difficult to cultivate. It took a very long time, and partially that's because once they were able to identify the full genome, it's actually a very small genome, and it's one of the only known pathogens that has a reduced genome. And so one of the characteristics of this is it actually has deficiencies in different types of metabolism, and so it's unable to synthesize certain amino acids, including histidine and arginine, and so there's this high reliance on the host. And so that's why originally it required culture within cells, and then once they were able to get the full genome, then they were able to create an enriched exenate culture media for growing this bacteria, and then since then it's been isolated from other different sites within humans. And then more recently, within the past 2 decades, because there was able to have the isolation of these bacteria, that's when there was started to be a better understanding about some of the immune mechanisms, specifically to the GI disease. And there have been mouse models of both GI and liver disease. And then only within the past decade has there starting to be a lot of growing evidence that not only is this associated with different pulmonary disease, but it can really be the causative agent of pneumonia. So then we're just going to transition to talking about a couple of different facts about Whipple's pneumonia. So there's kind of varying reports out there, but there was retrospective studies done in Asia and parts of Europe where they were taking different bowel specimens from individuals that had suspected respiratory infection or disease, finding that maybe about 3 to 6% of these individuals had Tropherma whippli within their lung. And within samples that were positive, about 15 to 28% of these, this was the sole microbe that they found within these samples. And most often, these cases were diagnosed as other sorts of diseases, such as TB reactivation, lung tumors, or pneumonias. Diagnosis of the lung infection does require either metagenomic sequencing or a pathogen-specific PCR of the bowel fluid. Also, if you have tissue, there's a variety of different histology stains that can be done, including periodic acid Schiff, Jimenez, also a silver stain. And so because of these different tools for diagnosis, it can frequently be missed due to a lack of targeted screening. Treatment can be really long. So it can require over 12 months of antibiotics, either oral antibiotics or a combination of IV and oral. And there is a high chance of treatment failure and relapse, especially in individuals that are treated for less than 12 months. There are a few kind of wide-scale studies that are looking at the impact of, or looking at T. Whippleii infection within the lung. But there was one study that was looking at T. Whippleii infection in people living with HIV that were undergoing antiretroviral therapy, or in HIV negative individuals. First, they were screening within the lung microbiome, comparing individuals that had HIV compared to those that were not. And overall, they saw that people living with HIV had higher carriage of this bacteria than those without HIV. And they also saw that if you looked at Trophema dominance within these individuals, again, over 50% of their lung comprising this sole microbe, again, they found that people living with HIV had more dominance than those without HIV. Now, because anytime you're doing studies of the lung microbiome, as I mentioned earlier, we know that the oral microbes do seed within the lung, but there's also the concern that you can have cross-contamination of the oral microbes when you are collecting the bowel specimens. So often you're using a paired oral swab or oral wash to then look at the microbes within that compartment as well. And they did this within this study as well, where then they were looking at the presence of Trophema within oral washes. And overall, they found they were unable to find Trophema in any of the samples that they collected, whether or not the individuals were people living with HIV or not. They next were looking at, within the group of people living with HIV, of whether or not there was any benefit of ART treatment on the carriage of Trophema. And they overall found that ART treatment was not able to enhance the control of Trophema whipplei colonization with the lung, and because overall the carriage was similar between those that were on ART or without ART, although they did note that they were able to follow a few individuals longitudinally before and after their ART treatment, and they did see that in some individuals the relative abundance did go down with ART treatment. So this is overall demonstrating kind of what does the Trophema infection or colonization look like within the human lung. And so next I'm going to just kind of switch back a little bit to talking then about what is known in the macaques. So then the next question you might be asking is, well, A, how do I know that this isn't contamination within my samples and whether or not it's actually happening within other macaque species? So there's been only a handful of other publications in other macaque species. There was this one study that was done at animals housed at the Oregon National Primate Research Center, where they were really more interested in looking at the lung microbiome in adult as well as aged animals. And they similarly found in about 2/3 of their adult animals that there was a dominance for Trophema, and that this decreased with age. And so this is overall suggesting, based kind of on the prevalence as well as that it's decreasing with age, that this is most likely a commensal bacteria. But then there's been other studies that have not been able to detect Trophema. So there was a study done in infant rhesus macaques at California where they were not able to detect it. One of the potential reasons why they were not is whether or not they were getting far enough into the lower airway. And then there was studies in cynomologous macaques from the University of Pittsburgh where they actually had varying different outcomes, where one of their studies had Trophema present and the others did not. So far our studies kind of fills in this gap, starting to look at the pigtail macaques. But it also started to make me think about that there could be different types of environmental drivers that may be leading to the establishment as well as the dominance of Trophema. So there can be a bunch of different factors that can be influencing the lung microbiome. I highlighted a little bit of this earlier within my talk. But these animals are going through different types of clinical and clinical as well as experimental histories. Some of them have had antibiotic usage. There's also variation in their external environments. So they were born at different locations, they moved around to different locations. We also have information about their animal demographics regarding age and sex and weight. And then there can also be changes to their local environment. And overall, these animals have very similar diets, so we don't think that this would be kind of a major contributor to driving the composition of the lung microbiome. But what we were kind of inspired by was what is known in the mirroring lung microbiome, is that overall, if you take animals that are born at different vendors, and then if you co-house them together, over a short period of time, you'll have convergence of their lung microbiomes. And this is just demonstrating that a lot of the drivers have to do with these environmental factors. And so we had hypothesized that environmental factors were also going to be primary contributors to microbial composition and diversity of the pigtail macaque lung. So it's important to know a little bit more about the environmental structure that these animals live in. So there's the colony of origin. This is where the animals are going to be born. And then from there, they may or may not move to different research facilities. There's different research wings, different research rooms, and then there can be different research cages. And this kind of lower part can really be a dynamic process throughout their lifetime. And luckily, we're able to capture all of this with our data. So we kind of wanted to start from a top-down approach of looking at these different factors to see which ones might be drivers of the microbial composition. So first, we started to look at the colony of origin on the left. So I'm highlighting several different research sites in which these animals were born, and overall, we saw no specific relationship between a specific site of where these animals came from in their microbial compositions. But when we started to look more about the place that these animals were living at the time of collection, that's when we started to see a little bit more differences. So most of these samples were collected here at the Washington National Primate Research Center, but some of them were also collected at the Tulane Biomedical Research Center. And so we started to see that the microbial composition of animals collected, samples collected from our animals here were divergent from those at Tulane. And then even within when we were looking at the specific research wing, again, the Tulane animals are in green. We started to see that there was differences between different research wings with certain ones having certain types of microbial compositions. So we then wanted to kind of go a step further, and that was to be looking at the different, the close contacts that these animals were having. So our animals, many of the ones that once they go into our different studies and infectious disease studies, they move into these Allentown cages. And what we're able to do is that you can have there's these dividers that can separate these different cages. And so we can either have what is called pair housing or run-through contact, where we can remove that divider and the animals can move between the cages. So essentially the animals can share the same airspace, or we can have grooming contact, where we set up the divider. They're still able to have social contact with each other, but are not sharing the same airspace. And so inherently within our data set, we happen to have 20 different animals in run-through contact. I'm now realizing that the left graph is not there, but it's going to be very similar to what was on the right, in which now I'm looking at the microbial composition of our lung microbiome, and the dotted lines are overall showing animals that are paired within the same cage. And what you can quickly see is that there actually happens to be really quite a lot of divergence between animals that are paired together. Oh, here it is now. And here it is with looking at a different beta diversity metric showing the exact same thing, that there's quite a lot of divergence. So another way to kind of look at that is that if we look at the amount of tropherma within the lungs of each of the animals and compare them, if there was a relationship, we would expect there to be a linear correlation, but overall we don't see that. And likewise, something that we can incorporate within our data is that some animals are paired for a very short time, and some animals are actually paired for over a year. So it could be possible that there needs to be a certain amount of time in order for us to have convergence of these microbiomes. And so one way to look at this is looking at the weighted unifrac distance, and that shorter distances are going to be more similar, and larger values are going to be more dissimilar. And if we look at overall of how many weeks the animals have been paired, again, we see no relationship with time. And so this is really showing that tropherma discordance is high and time dependent. And so what this is overall suggesting to us is that either we're not getting animal-to-animal transmission of this bacteria, or that there might be certain factors within the host that is leading to the elimination of having this bacteria being established within the lung. This kind of goes into the idea about how transient is the lung microbiome. There's not a lot of studies actually in healthy humans longitudinally looking at the stability of the lung microbiome. Most of them have been in the context of diseased states, but it's thought that it might be very highly transient. But if we if we look at data from another group that was looking at cynomolgus macaques, in this study what they were actually doing was they were looking at the lung microbiome over the course of simian immunodeficiency virus infection, and what they were mapping the animals for over a year, and what they were overall noting that even though there was kind of divergence across the different animals, that an individual animal actually stayed very consistent over time. And in this model, this is kind of also suggesting that even when animals are immunosuppressed, we actually don't get a lot of variation of tropherma over time, which is shown here in the red. So far we are thinking that there are certain drivers within the environment, but it doesn't seem to be that this is transmitting animal to animal. So next we wanted to look at the within a given animal, and this kind of now goes into our different paired specimens that we have. So for this, we're able to take our bowel specimens, our oral swabs, as well as our stool, and we're able to look at the microbial composition across these 3 different sites. And what this is overall showing is, if we just look on the left looking at observed features, this is how many different types of microbes are within these different compartments. And this is, again, demonstrating that the bowel and the oral swabs are going to have a lot less microbes than within the stool. But overall, the bowel and the oral swab are actually very similar in kind of the amount of bacteria that are within those. And this kind of goes across a lot of our other alpha diversity metrics, that the bowel and the oral swab are very similar, but are much more less complex than the stool. And so then we can then look at overall the composition of these different microbiomes across these 3 different locations within our paired specimens. And so here I'm showing in the dots is the bowel, and then we've got the oral swabs with the triangles, and then the stool within the squares. And what you can see is that each of these different compartments really does have distinct microbiomes. And we're going to be expanding this data set with more animals. But really the important question is whether or not we're actually getting any tropherma within these other sites that's leading to cross-seeding into the lung. And so far with our 10 paired specimens, we're only able to detect the tropherma within our lung. We don't detect it in the oral swabs or the stool. So this is suggesting that we're not most likely getting cross-contamination from other different sites. And overall, a lot of studies have looked at carriage of tropherma within the stool of different macaque species, including wild macaques, and it isn't commonly found within them, even though we do find it within the human stool. Now, I'm going to kind of talk about some of the conclusions of what we've done with this study so far, and then I'm going to be talking about the different types of future directions that we're moving towards in the future. So overall, we've identified that tropherma is a really highly prevalent, and it's bacteria, and it's really abundant within our pigtail macaque lungs, and that tropherma-dominant lungs have low microbial diversity and distinct community structures. Overall, we've started to identify different environmental drivers of the microbial composition and structure that may include the research site and wing, but not the colony of origin. And so far, we overall have poor evidence for tropherma transmission across anatomical sites or between animals. And so based off of the fact that not only do these animals have a lot of the different core microbes that are found in the human lung, but they're also harboring this bacteria that can become opportunistic within the human lung, we think that this can be a really highly and relevant and insightful model for studying the human lung microbiome. And so overall, this project has led us to have a lot more questions that we currently have answers for. And so we're approaching it from a variety of different perspectives and different questions that we have. So one thing is that we really do want to confirm, is trophema actually part of a healthy lung for these animals? We get such a wide range of the abundance within the lungs. We have some animals that don't have it at all, and some that are over 99%, yet all of these are considered healthy. I'll also say that looking at respiratory health and disease can be a little bit more challenging in animals, and so when they're alive. But what we're starting to do is that we are looking at the different immune profiles between animals that either have a non-dominant lung where less than 10% of their lungs are trophema in comparison to those that over are 90%, so that we can start to identify if there's any impact on the transcriptional signatures within the lung. And then we're also looking at pulmonary as well as peripheral inflammation to start to identify if there's any potential local or systemic biomarkers of this infection. We also have a major question about whether or not the presence of trophema in the microbial composition, whether or not this is influencing respiratory infection. And this goes back to why we originally started kind of looking at the lung microbiome. So I have an ongoing study right now that's actually looking at rhesus macaques, but what we've found is these animals were infected with SARS-CoV-2. And if we look at the level of trophema in the lungs prior to the SARS-CoV-2 infection, we see that animals that have more trophema with their lung overall have lower SARS-CoV-2 viral loads. And actually there was a recent publication also in Rhesus that was showing this same inverse relationship where higher trophema related to lower burdens for non-tuberculosis mycobacterium infection. So this is again further suggesting that most likely the presence of this microbe is somehow shaping the lung environment that is leading to different types of outcomes with different respiratory diseases. So we're looking at this in the context of SARS-CoV-2, but we also have samples from those that were eventually infected with influenza virus or valley fever infection, which is a fungal disease, so we can start to understand whether or not this is conserved across different respiratory pathogens. We also have collaborators at Seattle Children's as well as the Virginia Commonwealth University, and what we're working on is developing a customized delivery device where we can model the nasal interface and the lung of the pigtail macaques. And what we're working on is optimizing the delivery of dry powder inhaled antibiotics directly into the lung. So when we're able to do this, this will then allow us to modify the lung microbiome directly and look at the impact that this is having on the immune environment and overall respiratory health. We also are very interested on where is the source of the trophema across all of these different sites and how is it transmitted. Most of the data I was showing you today was from samples that were collected here at our Washington site or down at the Tulane Biomedical Research Center, but we also in our data set that we haven't sequenced yet that were collected at our Arizona breeding site at the Washington National Primate Research Center, as well as the New Iberia Research Center. And so by having these different data sets, this will help us to understand how different types of environments are shaping the microbial environment. And with this, continuing to have a bunch of different paired specimens so that we can identify if any of the sources are coming from our animals. We're also looking into different environmental sources that are present within where our animals are living to see if these could be potential sources for reseeding of these things. And then we're going to be continuing to study community dynamics and again looking at some of their demographics and their clinical histories to start to identify other things, including whether or not there might be an MHC component. We can track where the siblings we can track siblings to see whether or not there is also a relationship between MHC and trophema dominance. And lastly, something that we are undertaking is the arduous task of trying to isolate out this bacteria from the bowel samples. So far, we're 100 days into our culture. There's something in there right now, but we're kind of getting some different challenges. But one of our goals is that if we can isolate out this bacteria, not only can we then do whole genome sequencing on these different bacterial isolates so that we can really identify, is this actually Trophairoma whippli? And is it the same species that is found in humans? Because then this will start to elucidate why is it looking to be causing an asymptomatic colonization in non-human primates, whereas in humans, it can either actually cause asymptomatic colonization or it can lead to pathogenesis. And so by understanding more about this specific, the bacteria from these animals will help elucidate this. And then that will also allow us to potentially create other different models for studying the bacteria further. And so with that, I would like to thank the members of my laboratory, including Zoe within my lab, who's really been the leader of this project. I'd like to thank my collaborator, Steve Salaponte, who's done all the sequencing for us, as well as other individuals who have provided support and resources for this project. I'd also like to really thank the non-human primates at the different collection sites that provided these different samples, as well as my different funding sources. And with that, I would be happy to take any questions.
There's Lionel Matz. Thanks. That was wonderful. When I've heard talks about the microbiome, oftentimes, there is some specimen that's selected for the gut microbiome. It's often kind of fecal matter or something like that, which isn't exactly the microbiome at every centimeter of the GI tract. And like this, the BAL, which you seem to be relying on, is that which rinses out in the lavage, but it may not be the bugs that you care about that are in the exact compartments that are relevant. So maybe the Trophairoma is that which is easily shed, and maybe not as tenaciously stuck to the bad list. So how could you get at the compartments and where the actual things are? Are there experiments that you could or will have done that can show that they're relevant, that the bugs you can identify by the BAL are actually relevant in the place that they actually are in the living organs?
Yeah, so I can kind of address that a little bit. So at least in regards to the human microbiome, there have been studies where they've done serial bowels looking at different parts. And again, within a healthy human lung, it is pretty homogeneous, right? So if this is a healthy lung in the macaques, we could maybe assume that that would be the same. So something that we also have for some of our animals is that we do have paired blocks of tissue from the different lungs, and one of then our goals is that we can then be staining for the bacteria of these different compartments, and then we'll be able to kind of identify it that way. So yeah, one thing could be, because we do a bronchoscope guided bowel, you are able to go on one side versus another. We don't necessarily choose one specific side, and we haven't done paired bowel sampling across the two different lungs. So that would be a way to start to identify how much variance is there with these washes. Yeah.
Yeah. Yeah, that was a really nice talk, Megan. My question is sort of similar to the technical after you get the sample side. I always just kind of wonder, when you're amplifying all the different microbes, how you know that there aren't biases or skewing with primers or resampling, or are there any measures that are taken to try to get equal representation?
Yeah, so there are different parts of the 16S gene that can be amplified. We use amplification for the V3, V4 region, but others use other areas as well. So inherently, because of that, there will be some biases. Obviously, the main thing then is to stay consistent within the methods across all of your your samples. That's why sometimes trying to cross-compare microbiome data from two different groups can often be challenging. But the reason why we chose the V3, V4 region was that was the one that was commonly used for amplifying microbes within the oral cavity. And so we thought that those would be ones that would be best suited for these studies.
So you got an online question. I'm going to ask it in two parts. So the first is T-Whipple testing performed routinely in human bowel samples, and do your findings suggest that it should be?
So I don't think that it's, well, I'm not a clinician. I know that there are tests for doing it and PCR-based tests. I don't think it's routinely done, but I think that it is definitely an under-screened bacteria. And so I think that if people are suspicious of a bacterial lung infection, that it would be good to add this as a potential bacteria to screen for. Obviously, if individuals are diagnosed with a bacterial infection and are treated with antibiotics, it is possible that it would still clear out this infection as well, but you might not even know that you had it.
And then the second part is that one of the striking features of human Whipple disease is how variable the sites and the character of infection, so the brain and joints. Do macaques have similar diverse adaptation?
Yeah, so we've only looked at the 3 sites that I mentioned so far, the oral cavity, the gut, and the lung. But based off of the different tropism, for example, we've had interest in, since it can cause endocarditis, that could be, we could look at the lungs of our macaques. But we just haven't looked at those other compartments. But we do have a bunch of banked specimens for different animals where we know that they do have T. whippleI within their lung, and so that's something we can do in the future.
And then I have a question. Did I catch right in the beginning that you said that the organism is intracellular and that could? Yes. And so I guess I'm wondering how that plays into sort of the detection, right? Because if you're looking by sequencing, could you be detecting DNA that's associated intracellularly versus extracellularly? And, are they equally culturable if they're intracellular versus time?
Yeah, so what's known about it? What's known about the infection in macrophages is mostly in regards to the gut. It's not as well characterized of what it might be infecting within the lung. There's a variety of different alveolar macrophages or infiltrating macrophages that could potentially be targets of infection. And so there's going to be cell-associated bacteria as well as extracellular bacteria as well, which we're probably picking up in our bowel washes. But we don't really have a good understanding yet of what could be the potential, targets within the lung. What I will say though is that the one study that was looking at rhesus was that they were looking at kind of the composition of the cells within the lung, and they did find that trophama-dominant animals did have different levels of alveolar macrophages and other immune cells. So the infection itself could also be kind of shaping that immune environment to kind of then leading towards a more favorable environment and having more target cells and stuff like that. Okay, thank you everybody.